So thank you, Tim, for the introduction. I'm Jackie Surls, and it's nice to see some familiar faces. So it's very exciting to see some faces that I'm familiar with through video conference and names that I'm familiar with through email chains. So very excited to be here. And for those of you that I am not familiar with, I'm going to share a little video clip of our LuminexPLORE video. XMAP is the most trusted multiplexing technology for taking your research to the next level. By combining the expertise of Luminex scientists, our custom assay services enable highly sophisticated proteomic and genomic assay development. Our expertise covers a broad range of applications, including biomarkers, immunology, and infectious diseases, to name a few. We can also customize services to meet your research needs. Whether you're looking for custom coupling or full assay development, that's me. We can help your team reach its goals. So I'm really proud of this video because these are actual scientists that I work with personally. This is a few of the actual labs that we work in. So this is, not actors. This is real life, real lab, real people, my real friends and real colleagues. So this is, real people working on real projects, probably some of the projects that some of you are in here that we've worked on. This is a real manufacturing facility, that GMP facility that we're working on. That's actually one of my colleagues, Zach Garcia. So these are, real things that we do. So yeah, so that's just a little snippet of what we do. You can see the full-length video at our LuminexPLORE website. So some of the objectives that I want to cover today is who, what, when, and how. Who are we? What do we do? When can we help you? And how can we help you? So LuminexPLORE Lab is a custom services program that uses our leading industry XMAP technology to offer you a competitive edge for your scientific breakthroughs. And as you saw in the video, our expertise covers a broad range of scientific applications. So allergy testing, infectious disease, stem cells, neuroscience, and the like. And what I want you to envision is, thinking outside of the box. As you saw earlier, Pete's talk earlier from Bohr Biopharma, the SG RNA applications, thinking outside of the box of where you could use your XMAP technology. It's not just limited, to these applications. Where else can you use XMAP? Our mission and vision is to really empower you to explore using our technology outside of that box, and then really allowing, where we envision is giving you the support you need to be able to use this tool. It's a very powerful tool, but every now and then you might need that support. And so we want to be able to empower you and equip you to say, hey, I need help. Somebody provide you guidance and give you those tools that you need. So LuminexPLORE Lab is based in Austin, Texas. And if you ever want to ask your boss, can I visit Austin, Texas? I am happy to give you a tour and bring you on site. Here we have an arrow. Our lab is right there, right next to our R&D facility. The nice thing about being based in headquarters is that right across the street is our manufacturing facility. So whenever I need to get beads, I can just walk right across the street, ask our VP of manufacturing, may I have some more beads, and build you your custom kit. Also, the nice thing about being located in headquarters is adjacent to our lab is our training facility. So whenever you want to get training, and if you are allowed, obviously you guys are allowed to travel because you're here. This is where you would go to get training for on-site training, whether it be for instrumentation, custom assay development. This is where we offer the Luminex learning training courses. And so while you're there, you can come by to the Explore Lab and visit our lab for a tour. So here are our application scientists. Just to, we have a much larger lab, but these are our core team. We have myself, Carrie Whitmer, Ria Kang, Frank Medina. These are our on-site application scientists, and we have Steve Angeloni, which many of you are familiar with, who's probably been to many of your sites. And if he hasn't, please reach out to him. He has been instrumental making key contributions to Luminex. If you haven't heard of the XMAP cookbook, I encourage you to download it from the Luminex website. He has been a key contributor to making those protocols in that cookbook. These are our key core scientists. The nice thing about our Explore team is that they come from a highly academic background. So one of the differences between Luminex Explore and similar, people that are offering this type of service is that we have a very good understanding of your projects. We're highly academic, and then we're also hybrids of industry. So not only do we have that in-depth academic background, but a hybrid of industry, we have a very diverse team. So we have experts in biochemistry, experts in cellular and molecular biology, experts in infectious disease. So not only do we have experts in very niche fields, but then they're also cross-trained across the fields. So when they're assigned specialty projects, we handpick and hand select depending on what that project is. And then also supporting the Explore team, we have additional application scientists, associate scientists, we have a lab manager, project manager that also support this team. So what are your biggest challenges? So some of the things that we hear: can't get the assay working. So let's say it's something that you built and suddenly it's not working, or you had something that you built and all of a sudden it's broken and you don't know why. You don't have bandwidth. You need expertise for feasibility studies. Let's say you're working with a new matrix and you're not quite sure how to get that assay working in this new matrix. Let's say you were working with serum, now you're working with cell lysate, just as an example. There's no partner kits available that meet your needs. So, all the partner kits are for, all these other targets, and you have this really rare target for perhaps a really rare, porcine or non-human primate, and you're just like, how do I build this? You need custom coupling. All these things are things that we hear, and we're here to help and support you in those ways. And these are just to name a few. So here are some of the available services that we offer off-site and on-site. To the left, you'll see what we offer in our LuminexPLORE Lab. And I won't name them all, but just as an example, antibody pair screening, custom coupling. We do offer sample testing, custom assay development, assay conversion from ELISA to XMAP. And then we also do add-on validation options. For our FAS support on site, though we have advanced application services, we also have our FAS that can go on site to do 1-day training, 2-day training, 3-day trainings. And so it's very amenable and flexible to what your needs are. So basically, if you don't see it, just ask your sales representative, hey, these are my needs. How can you help me? So since launching about 2 years ago, there was an initial trend that we saw. So our initial offering was Let's just offer basic custom coupling. And as we heard feedback from our customers, there was new asks and new requests. And we saw growing needs and gaps that we didn't realize, or maybe we did realize, but we didn't fully understand the scope and need and how big that need was from you guys. So it started out initially with just basic custom coupling, and it grew to full-scale custom assay development of custom coupled beads, detection antibodies, reporter solution wash buffer, standards and controls, asking for reagent stability, lot-to-lot testing, sensitivity, specificity, all the things, right? Then it grew to GLP, GMP services, custom assay manufacturing, sample testing, involving our QC and QA team, and then having customers, you guys, come in and having an audit of our lab, which, you know, very quickly involved a lot of processes on our end to make all these things happen. So in short, basically, if you don't see a service offering on our menu, let your sales representative know or let me know. And if we have enough requests for those things, we can definitely add those things to our menu. So, you know, because custom assay development is one of our largest requests, I'm just going to go over this one specifically of what that development workflow looks like. So it is a phase-gated process where phase 1 is the project scope. And after that gate, those assay specifications, acceptance criteria, and design inputs will be clearly defined. During phase 2 is where the assay development will begin. And after that gate, the reagent selection, the matrix will be optimized, the assay will be optimized, and the assay performance parameters will be determined. All of those things will be locked in and identified. For phase 3 and phase 4, for fit for purpose, qualification, and validation, we'll make sure that the assay meets specifications, the acceptance criteria, and that the design outputs are defined. For phase 5, if you desire that Luminex manufactures it, which we can do it non-GMP, we can do it GMP, or we can design transfer it back to you or to another entity. If we have Luminex manufacture it, at that gate 5, we'll make sure that we're capable of producing a passing lot at scale, and then we'll make sure that we deliver that product back to you. So I'm going to give you a few examples of a few scenarios of what this could look like, of a few case scenarios. So a case scenario of custom coupling. So for this scope of work, what it could look like is custom coupling of antibodies or proteins to magplex microspheres at small scale or large scale couplings. We would do QC coupling confirmation to make sure what we coupled onto the bead is indeed there. And the deliverables would be protocols, data files, and your custom coupled beads. In the second case scenario would be custom assay development. The deliverables could be 2 options. And this is not limited, but this is just an example. Protocols, data files, and a prototype small-scale build for you to validate on your end. Option B would be protocols, data files, and a first custom manufactured material lot, which would be the standards, controls, beads, detection, buffers, and reporter solution. So how would you get started to begin this process? One way would be contact your sales representative if you know who they are. Second way would be go to our website at www.luminexcorp.com /LuminexExplore. or go to our website and click on the link that says Submit Custom Service Request, Step 1. Step 2, you would consult with an application scientist. And then Step 3, you would begin assay development with our Luminex expert.